Welcome back to the course on functional genomics. So, let us look into this scenario. For example, you want to look at, the question is that you have a cancer tumor tissue and you have the same normal tissue from where the tumor has grown. Now, you want to look at, in the tumor condition, what are the genes that are upregulated, whose level, expression level has gone up, which could possibly is involved in some tumor formation. And likewise, what are the genes that are normally expressed in the normal cells, but now shut down in the tumor cell. That means that that gene is required for the normal cell function, right. So, what do you do? So, you have these two samples, you extract the mRNA from each one of them, and then you make what is called as a cDNA. That is something that we discussed. So, you basically make a complementary or copied DNA. And when you make the cDNA, what you do is you tag the DNA being made with certain fluorescent moieties. So, let us say I am going to use a fluorescent moiety that gives red fluorescence when I excite with certain wavelength for the cDNA that derived from the cancerous cell, or I can use, for example, for the normal cell I am going to use a fluorescent moiety that gives you green color if I excite with a particular laser or a wavelength of light. Now, what I do is, now I am going to take equal ratio of these two cDNA, say for example 2 microgram each, I am going to mix, and then this is my gene chip, something that shown here. Now, I am going to hybridize, I am going to allow this cDNA, because now I have got rid of the RNA from here. Now, we have only one single stranded cDNA, which is complementary to the sequence that are spotted there. If if there are, I have 10 copies of a given gene spotted on a chip, and if the expression of the gene is same in both cancer and normal cell, then going by the probability 5 copies of the gene will be bound by the cDNA derived from the tumor cells, and 5 copies of the target sequence will be bound by the cDNA derived from your cancerous cell, right. So, that is where it is. But if it is the equilibrium shifted, you have more of that transcript present in cancer cells, you are going to have more of cDNA that is going to go and bind to the spot. So, by looking at the relative ratio of these two fluorescence, I will be able to tell whether the expression level is altered or not altered. That is what is shown here. For example, this is your array, you have spots there. So, what I am doing is that once these are arrays hybridized, then I am going to quantify the fluorescence by exposing that chip to a laser source, excite that fluorescent moieties, therefore it emit lights, and I am going to measure that. So, what I have done is I have first measured for the green and measured for the red. I am going to look into the relative expression of red and green, which will tell me whether, as compared to the normal cell, in the cancerous cell, whether the expression level has gone up or gone down or not expressed, and this is how you are going to do. So, let us see that this is what it is. So, if the expression level remains same, pretty even between the two source, sample A and sample B, then I would get something like green, yellow, because this is a mixture of red and green, right. Whereas, the expression level in sample A for a given gene is very high, I am going to get a red fluorescence, because the green fluorescence coming from sample B is going to be extremely low, because it is a competitive hybridization. Say suppose for every one transcript for a given gene from sample B, you have 9 transcript from sample A, and I have 10 targets here. So, 9 of them will be bound with the cDNA derived from sample A. So, you are going to have more of red color, that is what it is. On the other hand, if it is reverse, 9 here and 1 here, I am going to get more of green. So, by looking at the intensity of these two fluorochrome, the light emitting moiety, I am able to interpret whether the expression has gone down, gone up, remained the same, right. So, this is the way you are able to do that, but you are not doing it just for one gene. In one go, you are able to do it for thousands and thousands of the gene with little amount of RNA. So, that is the power of the microarray, and which has really changed the way we look at the gene expressions, and this is one of the landmark papers, perhaps the first paper that has looked at the expression in a genome-wide basis. This came in 1997, where they looked at the yeast, the popular model, and its expression analysis, what is called as genome-wide. You can see that now this is being more often used, but that is the first time they have used. So, genome-wide expression studies. So, what they have looked at? So, they have looked at relative expression of genes if of yeast that were grown in two different medium. One is glucose, or the other one is galactose. Okay, and they looked at what are the genes that are turned on if you grow the yeast in glucose, and what are the genes that are turned on if you grow them on galactose. So, they looked at how the profile, expression profile of all the genes changes simply by modulating the culture condition. And how many genes? They have looked at close to 2,500 genes in 1997. It is a huge shift from the way people are looking at it. And then it is not only expression, but they are looking at relative expression. That is what the power of microarray. So, it is not just saying expressed or not expressed, but whether the expression level is higher as compared to this condition or lower as compared to this condition. And you can see that there are dots that are completely green, there are dots that are completely red, but majority of yellow, suggesting that majority genes pretty much did not change in the expression, but some of them shut down, some of them activated, and so on. So, that is the first report on the application of microarrays for gene expression at the whole genome level, came in 1997. Since then, there are a large number of papers. But what is interesting here is this kind of analysis gives you is, now you have not really looked at what gene it is. You have spotted every damn gene that you have it, and you are going to look at whether the RNA is made or not made. Now, having found a difference now that they are overexpressed or underexpressed, now you can go and look at what is the function of the gene and what is the pathway, and then how that may make a sense, because this is after all a data, you have to analyze this. So, this is what they are showing here. For example, there are proteins, if you give a heat shock, for example, whether amino acid metabolism is altered, whether the gene that are gone down, gone up, and so on. So, this is a kind of post analysis which gives you much larger picture, which can help you to come up with a newer hypothesis to test the function. So, that is another paper in 1999. Again, it is a landmark paper. They have looked at cancer, because one of the biggest challenge in cancer is that you have different grades of cancer, subtypes of cancer. So, your treatment basically depends on if you are able to diagnose which subtype of cancer that one person is suffering from. And there is always a challenge as to how do you classify. So, most often people use histology looking at, but these are much, the changes that you see over there is very late. By then, cancer has really progressed to much severe level. But if you are able to type it early, it is better. So, to type that, can you look at the expression profile? Can you look at what are the genes that are expressed? Can you type them, that is this type 1 not type 2 and vice versa, and then one can come up with certain therapeutic modules. And here what they have done is they again take a approach for cancer classification based on gene expression. They are monitoring DNA microarrays. This they have used this microarray to look at the gene expression, and then what they have looked at is that leukemia is the model, and they have looked at two types: acute myeloid leukemia, acute lymphoblastic leukemia. These are subtypes, and they are able to show, for example, there are certain genes that are present or expressed in a very distinct way as compared to AML. ALL is the acute lymphoblastic leukemia, AML is myeloid leukemia. So, they are able to type now. So, this way they are able to tell that this tumor belongs to which subtype. If there are drugs that would be more effective in AML, this person can straight away be given AML without really waiting for further confirmation. So, this is again a landmark paper which really helped us to classify the tumor and go on. And if you look into the PubMed again, if you give the query as whole genome microarray, and you can see that how many studies are there, right, and it is only growing. With time it is going to go up, and that is the application. And this also brings in a challenge. The challenge is, what does that mean? So, you are spotting all the genes that are predicted or identified in the genome. For many, you do not really know what is the function, right. So, if you get some change as the microarray suggests that the expression level gone up or gone down, what really it would mean. So, this really requires not a manual analysis, it requires computational tools, and that is where the researchers have found a need that all this data that comes out of the microarray, the fold difference in the expression level and so on, should be deposited in a common repository, because even the tools to analyze them are evolving. So, if the data is present, then one can evolve tools to analyze that. So, what is shown on the screen is one of the databases called Geo Omnibus, which is nothing but gene expression omnibus, and this is a database wherein people deposit their expression analysis data there, and anybody can use it. So, that led to a new branch of bioinformatics called as gene ontology, meaning what does it mean? So, say suppose I am getting a gene whose expression level is altered, what does it really mean in terms of the biology? Because after all I am looking at biology as to how that gene has altered the function, for example cancer progression, or it's whether a drug is able to whether the tumor is able to respond to the drug or not. So, all these people who are involved in such kind of high throughput genome-wide microarray analysis, they came together and sort of came up with an idea that you have to have some tools to classify the information that you get out of microarray, and that led to a very powerful bioinformatic tool called gene ontology, and that now it is being widely used not only for human. You can see that it is for whether it is a mouse or yeast or Drosophila. All these people came together and came up with a uniform way of classifying genes based on the function, and this is a dynamic process because the gene being studied today and you know that this is involved in this function, the database is updated. So, as the results come out, this database is being constantly updated, and this is called as the Gene Ontology Consortium or Gene Ontology. So, that is the website that all of you can go and look at, and when they formed this consortium, this is the mandate. It says, I read it out, genomic sequencing has made it clear that a large fraction of the genes specifying the core biological functions are shared by all eukaryotes, whether it is the DNA polymerase, histone protein, they all do the same function whether they are looking at yeast or the human. Knowledge of the biological role of such shared proteins in one organism can often be transferred to other organism, because if you are looking at a developmental process, you will never be able to study that in human, but you can study it in zebrafish or fly, and that information should help us to understand the genes that are present in the humans. The goal of the Gene Ontology Consortium is to produce a dynamic, meaning dynamic, it has to be updated every day because the research is going on. Controlled vocabulary should not be something that everyone uses a different terminology, it should be the same if it defines the same function, therefore easy to understand, that can be applied to all eukaryotes even as knowledge of the gene and protein roles of the cell is accumulating and changing, because you have to have some unifying code. To this end, 3 independent ontologies accessible on the World Wide Web, that is geneontology.org, are being constructed. That is when they put this proposal. Now it is pretty much is being used. These ontologies are grouped into 3 distinct domains. One is biological process, second is molecular function, cellular compartment. For example, I may be looking at a protein which is a transcription factor. So, the function of the protein is DNA binding. Where it should express? It should be present in this compartment which is nucleus. So, this protein gives you 2 information. 1, it is present in the nucleus. 2, it is DNA binding in its activity. What is the third function? It may be involved in skeletal development. So, that is the process, right. So, this would evolve as people are studying, it will evolve. So, they are keeping on updating this database. So, when I do a microarray for a given tissue or whatever process, I found a gene, this is the gene whose expression level is very high, then immediately I want to know what does it mean, level gone up, what does it mean. So, this is a protein that is present in the nucleus, therefore, the protein level should be high. It is a DNA binding protein, and therefore it should be activating or inactivating a transcription, and what process it may affect, for example skeletal development, right. So, this is how you look at it. So, this is what it is shown, and each theme has sub nodes, because that within molecular function you could have enzymes, you could many things, biological process likewise we will see some of them, compartment it could be nucleus, within that it could be membrane and others so on. So, they have classified it. So, as the information evolves it gets updated. I would urge you that you go to this Ontology website and have a look at it. I am going to give you examples of 3 case studies. We are not going into why this study was done. If you are interested in, you can go and look at this particular paper. What I am showing is how they have analyzed the data. Let us look at, they have done a microarray between 2 different samples looked at, and there are certain genes whose expression level came down, whose expression level went up, and they are looking at what is called as biological process, right. What is the biological process? Sensory perception. 3.7% of the genes whose expression level came down are involved in sensory perception like touch or smell, whatever. Protein oligomerization, proteasomal ubiquitin dependent protein catabolic process. These are the genes whose protein involved in proteolytic process. Regulation of cell proliferation, controlling cell division, angiogenesis meaning blood vessel formation, cell proliferation, synaptic transmission. You can see that how we can say that, okay, is it that? It is very interesting. Almost close to 20% of genes whose expression level are upregulated are involved in multicellular organismal development. That means some growth happening, right. You can go back now, look at the paper, what did they do, why possibly overexpression of the gene led to that process. It will be interesting. And you see that there are down regulated transcript, regulation, transcription, DNA dependent and so on. So, this is how you can classify. Molecular function, again this is a second sub theme. Then you see that there are some are kinase activity, some are transcription factor, ligase activity, GTP binding, DNA binding and so on. This is what is the specific function of the protein, but the pathway could be different, but we are looking at the specific function. Or you can go at the level of cell as to where it is located. For example, these are present in microtubule, nucleoplasm, Golgi apparatus, cytoplasm, nucleus, and you have information. So, as it is evolving, if somebody has done in yeast and you have a protein which is a homolog, meaning the protein that represents the yeast gene in the human, it is likely that it does function. So, it is an indicative, it is not saying that that is absolutely that is the function, but it is indicative. That is how you go and further model it and test it in the system. So, anybody is working on any system, now everything is put on the website, linked to that gene and protein, and you are able to get, without really getting into the paper, you are looking at thousands and thousands of the papers that are there whose results are sort of linked to all these genes. You do a microarray, you get all this data. So, it is a powerful tool made possible by using bioinformatic approach, and that is being constantly updated by looking into the literature. Again, there are some bit of human element involved, it is not everything is automated, but certainly one group of people are doing it for the benefit of a large number of scientific community, right. So, the microarrays not only revolutionized the study of large scale mRNA expression, that is what we discussed, but also has changed the way the genome was analyzed, right. There are changes at the genome level. For example, there could be repeat regions, repeat numbers and increase, or I could have a small segment of the genome is duplicated, 3 copies and so on. So, what you have looked at is a transcript profiling. We have so far looked at the mRNA, but now you have such kind of arrays for non-coding RNAs and microRNA and so on. The other thing is to look at the changes in the DNA that makes a gene to be active or inactive. This is called as chromatin immunoprecipitation, something we will discuss little later. And there are also arrays to understand what is called as comparative genome hybridization. For example, whether a region of a genome, whether that has got duplicated, the copy number increased or lost, that can be looked at. It is involved in large number of pathology including cancer, we will study little later, and of course sequence variation, what is called as single nucleotide variations, large scale, and also alternative splicing. We discussed that each gene has multiple splice forms. Now you have arrays that can detect all these things. So, before you end this particular lecture, we will look into one last approach. Again, we come back to this our real reverse transcriptase polymerase chain reaction, and in this what we are going to do is microarray is indicative of difference in the expression, but if you really want to quantify, people have to come and look at individual genes to quantify them. So, this PCR we already discussed, you are going to amplify a segment of cDNA, but that does not give you the difference. So, you want to really quantify the difference of the expression level between two different samples for the same mRNA. Nowadays, we use a technique called as quantitative PCR or real time PCR. Here it is the same principle as like a PCR. One of the ways by which we can detect is to look for the number of the duplex DNA being made, because more the number of duplex DNA in a given number of cycle, that means that you have had more copies to begin with. So, if you are comparing two different samples, then you will be able to tell whether you have had more transcript as compared to the other. The idea is that you have what is called as one of the ways by which you study is you have the probes, which are what is shown here. The probes bind to your single stranded DNA and they have certain fluorochromes, and the fluorochromes are linked to that probe because it is you have the oligonucleotide, these fluorochromes are there. Normally when they are together they will not fluoresce, but when in the bound form, if the DNA polymerase come and dislodge them, in that process they sort of break them, then you get the fluorescence. That would tell you how many of them are bound, how many of them are converted. That would give you a relative estimate as to how many copies you started with. With that you can identify, measure the relative difference in the transcript. So, this is a very accurate way by which you measure the transcript, and that is pretty much the end of this lecture, where we looked into how people study large scale analysis of the transcripts, and in the next lecture we will move on to look at how we study the genome. Again we will go back to the genome and its dynamics, significance, how does it affect the transcription.